10 o'clock past. Yeah. Should we start now?
Yeah.
So because it is past 10 o'clock, and I think we can start.
Yeah, I think we can start.
Okay, sir.
Yeah.
So our first talk today is again by Professor Nmr Jagannathan, whom I know very closely as the first president, the founder president of the Indian chapter of International Society of Magnetic Resonance in Medicine. He is the fellow of several societies. He is the fellow of the International Society of Magnetic Resonance in Medicine as well. He is in the board of and as chairman of several of the scientific bodies of the government of India, including the one in which I am also there, that is Indian MRI project. The Indian MRI project by the Ministry of Science, the Ministry of Electronics and Communication is now ongoing, and we hope that soon an Indian MRI will be there. The cost of MR will come down, and it will be useful for several people. So we are all waiting for that MR to come in. So I'm not going to introduce Jagannathan sir more than this because he has been introduced yesterday. Let us hear his talk on applications of in vivo MR spectroscopy in cancer research. Sir.
Thank you, Professor Kesav. It was very nice, and thanks once again the organizers, Dr. Sunil, Vinesh, Vijayan, and others. And my friend, Professor Bhatt, also is there. I wish him good morning. And let me now share it.
Good morning.
Good morning. Somehow, Vinesh, I used to make some errors. I don't know where I do that. The full screen I put it. Yesterday also I goofed up.
No. You are right. That is fine, sir. I mean, when you go to full screen, because you are seeing your own window, the Google Chrome window. But then in the bottom taskbar, you have the PowerPoint there. I could see it. You go to there, click in.
Yeah.
And then full screen now.
Are you able to see this full screen?
Yes, sir. Yes, sir.
Yes,
okay, good. Thank you. So once again, thanks for the organizers for inviting me for this nice meeting, which I have also been attending on and off, and I find all the lectures are very useful, and I hope for the participants it was more useful. Now today, myself and later Professor Kesav would be talking about the application of in vivo MR spectroscopy in cancer research. I think Dr. Kesav's talk is a little bit more involving MRI as well as MR spectroscopy. Today, this particular talk is basically on the use of NMR spectroscopy in a living system. That is what I'm going to do that. Yesterday also I showed this slide to you. The application of nuclear magnetic resonance in the earlier days, basically, if you are confining yourself to the application in biology, it is for determination of the structure of macromolecules, conformation, function, and dynamics. And yesterday, I talked about the application of NMR in biology, the other aspect of imaging and in vivo spectroscopy. Today, I confined myself to the imaging. Today, I will talk about the in vivo MR spectroscopy, how this in vivo MR spectroscopy can be, in addition to MRI, will be useful for clinical diagnosis. And more than the clinical diagnosis, the in vivo spectroscopy basically gives you an indication of the physiology and the metabolism of the tumor. Remember one thing, that when a cell becomes malignant, everything has to happen at the cellular and molecular level. So if you can understand about the disease process at the cellular and molecular level by a tool like NMR spectroscopy, then you are at a very great platform to know about the tumor. One has to understand the tumor physiology and the metabolism if one has to devise a drug or design a drug. And yesterday I pointed out how MRI, the imaging aspect of the medical MR is useful for diagnostic purpose and as well as for assessment of the tumor response. During the assessment of tumor response, I showed a couple of slides on breast cancer and prostate cancer because that two areas, the one that I concentrated the last 30 years, is that you see the reduction in the tumor size visually. Otherwise, what clinicians normally do is that they resort to the normal biochemical test, ultrasound or CT or PET. But here, you actually not only see the tumor size being reduced if patients respond to a particular treatment regimen, then you can also do, along with the MRI, the in vivo spectroscopy to understand the physiology and metabolism of a tumor responding to the treatment and also a tumor not responding to the tumor. Both these issues, which MR spectroscopy, in vivo MR spectroscopy gives, is very useful for our understanding of the disease process. Now, just to recapitulate, you people have been pounded with this kind of information for the last 3 days. What I have shown on the left panel is a free induction decay, which is the normal NMR signal. I know, I hope that you know what you mean by FID, free induction decay. Now, this is the NMR signal, which is the time response of the spin system. So when you want to study how many molecules are present in a sample, then you need to Fourier transform this time response. It is done by a Fourier transformation. And if you have only one single, say for example, water in your sample tube, then you get a nice signal. But if there are many other signals, like for example, I yesterday made a lot of statements, number of times the statement that MRI is basically an imaging which is nothing but a two-dimensional or three-dimensional pictorial representation of the hydrogen density that are present in human tissues in various organs. That is what is MRI in a nutshell. Now, in MR, I also made a statement that these MR images are hydrogen images or proton images. They arise from the water, the protons present in the water and fat. So if you have water and the fat, then your free induction decay will look like what is under D on the left side corner. Because there are hydrogens in water. Hydrogen in water is an aqueous medium, whereas the hydrogens in fat are not in the real sense of aqueous medium, because fat has a lipid chain. So the hydrogens are in a different environment in water and in fat. So you have two FIDs, like the one I have shown in A, left A, which are superimposed upon one another, but there are frequency wise that are shifted. So that is why you get a beat pattern. Two FIDs superimposed one upon the other with a frequency shift, which when you Fourier transform, you get the two signals, one, the rightmost one to the fat and the other one to the water one. So if you want to have a little complex system like ethanol, then you have a NMR spectrum that I have shown on the right side, one for the methyl group of protons, methylene protons, and the hydroxyl protons. So this is what is called as a chemical shift, which has been introduced by earlier speakers, and that is what you do in a normal NMR. Try to identify the proton positions or any nucleic position and try to assign those lines, and from there you talk about the molecular structure. So what is the information that you basically get from NMR spectrum? For the sake of in vivo spectroscopy that I'm going to talk, I'm not going to do the structure determination that you are being taught in all these days, but now because we are doing this on a live human being or a living organism, you don't have time to go to NMR and other NMRs to find out what are all the different resonances or metabolites or biochemicals present. So you have only a spectrum that is available. It is shown like one. So what you basically measure is a chemical shift, then the line weight, then the intensity, which is the amplitude. With these limited information that you get from an in vivo spectrum, I am stressing the point that that is what we measure in an in vivo spectrum, not like the NMR spectrum where you can determine the concentration, the intensity and other things, which I will not go into the details. So basically you determine the chemical shift, the intensity, the area under the curve, or the amplitude, and the coupling constant if you can measure it. Okay, so this is the kind of a spectrum that Jeet and Vinesh has introduced to you. Very complex spectrum left up corner is a 1D spectrum of a 130 residue protein. It contains thousands of protein. Naturally, you have transons or lines. So, one-dimensional NMR cannot decipher which resonance is which is due to which amino acids and which are coupled to that. So, you do 2D NMR and 3D NMR to determine the structure. So, this is what Jeet has introduced in your. Now, let us now discuss what is NMR spectroscopy that you are familiar in doing with a sample, how it can be applied in real sense of a human. Yesterday, I talked to you about MR imaging, and I have been making stress on the point that it gives structural information. What I mean by structure is that you can identify the different anatomical regions of the organ that you are imaging. Yesterday, I also showed you a sagittal image of a brain, wherein I have marked in each of them where is the cerebellum, where is the frontal lobe, where is the pons, where is hypothalamus, everything. That means you are, through the structural information, you are getting the complete morphology and anatomical description of the organ that you imaged. I made a statement that it is superior soft tissue contrast and it has a multiplanar imaging capability, and then functional information can also be obtained through functional MRI. Let me, for the general audience, also give you one kind of a message. Functional MRI means it does not confine itself for us to study the cognitive functions of a brain. MR angiography is a functional MRI. Perfusion weighted imaging, diffusion weighted imaging that I showed yesterday are also functional MRI. So also the dynamic contrast energy. Anything that you do as a time function is a functional MRI. Okay, but then generally functional MRI, without adding any add-on to it, people say that it is due to brain functions that we are doing. It is okay in a general term, but specifically if you argue functional MRI includes other images like angiography, perfusion, diffusion, and dynamic contrast. So basically MR imaging gives where is the tumor in an organ. Now, if we combine MRI with MR spectroscopy, then you get the metabolic information. It is very useful to study the physiology and metabolism. What I mean by metabolic information is that if you look at the right top corner, where I have shown the brain image of a patient suffering from a brain tumor, which is at the middle. You can see the white portion, which is a tumor. Now, if I put a voxel means volume element or region of interest of a square, say for example, 10 millimeter by 10 millimeter by 10 millimeter or 20 by 20 by 20, which is 8 ml voxel. Then I will get a spectrum from the normal tissues, which is the bottom of the top right side corner of a normal brain tissue. It is dominated by 3 peaks. Now, if I wanted to study the second aspect on the left side that I have given under MR spectroscopy, the physiology and metabolism, then I shift the voxel from the normal portion to the tumor side, wherein I see not the normal kind of a spectrum that you see here, but lipid, the absence of N-acetyl aspartate, creatine, which was very high in the normal brain tissue, is much less, and choline is very high, which was less in the normal tissue. That means you are actually studying the physiology and the metabolism of the tumor, which would be very useful if you do a longitudinal study with a large cohort to determine the efficacy of drugs if different groups of patients are given different kinds of treatment regimen. That means you also have a tool to monitor the dynamics and pharmacokinetics of the drugs. And then I also told you, like MRI, you can also use MR spectroscopy to monitor the treatment. You can, like MRI, yesterday I told you, the right bottom corner, role of in vivo MRS in clinical medicine, like MRI, is same, like diagnosing it and differentiation between malignant and benign lesion. Identify a small lesion and try to study the physiology and metabolism by doing spectroscopy. You can also get biopsy like MRI and treatment response. So in the initial days of MR spectroscopy, evolving under the various researchers who were doing human studies, there are 3 major steps that took place during the evolution of in vivo MRS in clinics. The first one was the surface coil. If you look at the bottom left side, I have a surface coil, which is the black filled circle, which is the surface coil. As the name implies, this surface coil can be placed in the region of interest that you are going to have a spectrum from it. So, suppose a person has a tumor in the temple region over here, then you put the surface coil over the temple region here, and then you excite the region, which is directly proportional to the diameter of the surface coil, and then get a spectrum. Like, for example, mainly the initial days it was phosphorus spectroscopy, like the one that I have shown from the right side bottom corner. So the surface coil is the initial invention in the in vivo aspect, and it gives only information of the tumors which are very close to the surface of the human organ. Remember one thing, when I do this kind of an MR spectroscopy, the physics law says that the excitable region is the dotted region on the left side, which is proportional to the diameter of it. That means any tissue that is within the diameter, I will be exciting it. That means you are going to pick up signal not only from the tumor tissue, but also from the normal tissue. Anyway, let us come to that little later. So the right bottom corner, if you see the phosphorus spectrum, basically this phosphorus spectrum gives you the complete energy metabolism of the cells in that particular region from which I got the spectrum. It is the human head temple region. You can see all the three ATPs, alpha, beta, gamma. Phosphorus basically gives the energy window of the cells, and phosphocreatine, phosphodiester, inorganic phosphate, etc., etc. Now, the next one is the single voxel spectroscopy. I will explain to you a little later, and the multivoxel spectroscopy, which is the latest one, which is widely used. So now, sticking on to the surface coil spectroscopy, we also did a lot of work in the initial days with the surface coil, and this left side panel actually gives the P31 MR spectrum of a breast tumor. I have not, for some reason, shown the normal breast tissue phosphorus NMR spectroscopy. But it is suffixed to say, and taken from me, that the PME, which is very higher, was much lower in a normal tissue. That means now, in addition to having the sort of a concentration or the relative areas of the intensity of the various peaks of NTP, which is the same as ATP, then PDEPA, now I can talk about the metabolic pathway involved in the cancer cell in the breast tumor, which is given in the right side panel of my slide, wherein you see the metabolic pathway. You can now identify most of the energy metabolism of this tumor cells in this one. And in this particular breast cancer, I'm now telling you that PME is much higher than in the normal. But if I do the same thing in other tumors, for example, in the brain pathologies, this is what the literature actually says. I have summarized this one. If you take stroke, PCR, phosphocreatine is less, ATP is less, whereas PA is very high. And pH is initially in most of the tumors is less, and later it increases. If you take glial tumors, PCR less, PA is increased, pH is increased, PD is decreased, PME. Like that, you can actually find out. Say, for example, in Alzheimer's disease, increased PCR, decreased PA, increased PDE and PME. So one can, these metabolite, phosphorus metabolite, phosphate metabolites, you can now study the metabolic pathway. So basically, this in vivo spectroscopy gives you a tool which can be interpreted in the metabolic pathway of the tumor. And then you can study about the tumor metabolism. But unfortunately, the surface coil spectroscopy has a lot of limitations. I already gave you a couple of examples of what are the disadvantages of the surface coil spectroscopy. See the left side bottom corner. The surface coil is about, say, 10 centimeters. That means it will excite 5 centimeters of the temple region of the human head. That means I'm going to pick up all the normal tissue as well as the malignant cells. If the whole tumor is occupying, then you can take faithful reproduction of the tumor phosphorus spectroscopy. But if the tumor is confined only to 2 to 3 centimeters and the rest of the area is normal, then you are at a contamination. And another disadvantage is that if there is a tumor which is deep inside the organ, whether it is head, breast, prostate, human body, organ, abdomen, it doesn't matter. Then you cannot do that. Because the excitable, according to the NMR physics, is that excitability of the RF is limited by the diameter of the surface coil. Maximum I can have 10-15 centimeters. So that means diameter 15 centimeters means 7.5 centimeters. That is the only depth that I can actually go. More than that, it will become normal and abnormal tissue. So it has a lot of advantages. So people started to look for doing localized in vivo spectroscopy. What do you mean by localized? Yesterday, when I gave a talk, after that, there was one gentleman who asked a question whether I can excite at the simultaneously all the three gradients. I said, yes, you can do it and get a volume. That is exactly what we do in vivo spectroscopy. But most of the in vivo spectroscopy, remember one thing, are all image guided. That means you need to do the MR imaging of the particular organ that you are interested in to get a spectroscopy initially. Then what you do basically is that this is the paradigm that I have given. You look at the top portion of the slide. Patient positioning in the breast coil, global imaging. I'm giving an example of a breast because that was my area of specialization. And then you do for 10 to 15 minutes MRI and localize the tumor like what I have shown, red one in the middle of that tone. This is the breast tumor over here. Then we had to find out, yesterday I also gave an example of a dynamic contrast agent given to the patient to find out whether the tumor is benign or malignant. Most of the times when we handle a very big tumor, we are sure that it is going to be malignant. So sometimes we do away with the dynamic contrast while doing spectroscopy. But in very small tumors, we wanted to find out. That means we need DCE and then do the time profile curve that yesterday I talked about. We identify that it's a malignant. Put a voxel now. Voxel is volume element. And then do the shimming. Like Vinesh was finding out on the first day, that shimming is very, very necessary if you do a spectroscopy. Why shimming is so much necessary compared to when you are doing imaging is that you need to find out the various biochemicals or the metabolites that are present in the human tissues, which are distributed in a chemical shift axis. As has been pointed out by earlier speakers, that if you have hundreds of lines, then what you do? You do a 2D NMR, provided first your field homogeneity is homogeneous, because you need to separate out, say for example, the proton from an alanine from one to the threonine methyl group or the other one. So you need a very high homogeneous field. So what we undo for imaging by applying a gradient. Yesterday I showed a human head, 3 points that I want to image, and if I put a gradient, then I can find out the location of the 3 points. Whereas if I don't have a gradient, then all of them will give the same signal. I even explained to you with the 2 tubes of water. So that is for imaging, you have a gradient. Now you want to undo that and do a voxel shimming so that your field over this voxel region, this red square, is uniform so that you can find out the small subtle difference in the chemical shift between the various metabolites that are present in this tissue. Then you do the particular sequence, which I will explain a little later, but not in much detail, with water and lipid separation, like what I have shown in the right bottom. In breast cancer, water and fat is normal because breast contains most of the fibrogranular tissues, adipose tissues. So you have to suppress this to see the biochemical E. coli. But if you do, say, for example, in brain, then you have much rich biochemicals, neurobiochemicals, which you can monitor. So it depends upon the organ that you investigate. I will show you some examples of the breast and prostate little later, but first take you to how the MR, in vivo MR is done. Suppose I want to get a spectrum from the thalamus region of the brain, the red voxel that I have shown on the left side of this slide. Then I do the experiment exactly like what Dinesh Vijayan and Jitendra Chug will do it. Get a spectrum without any water suppression. You know that the metabolites that are present in human tissues are in the millimolar or micromolar range. compared to the concentration of the water, which is roughly about 50 molar concentration. So in the presence of the huge water, you don't see the various neurobiochemicals that I have shown at the bottom here, like N-acetyl aspartate, creatine, choline, myoinositol, etc. Reason is that the water is in huge concentration. So you need to have method in which you suppress the water. Once you suppress the water, you see various biochemicals that are present. Now, these neurobiochemicals that are given to us by the in vivo spectroscopy, how is it useful for me to interpret the status of the tissues or the metabolism of the tissues? You see what is on the right side. This is the Krebs cycle. This is the brain cell metabolism that is happening. So whatever I have marked with the different color, like glycosylphosphocholine, phosphocholine, N-acetyl aspartate, lactate, PCR, glutamine, glutamate, etc., are all seen here. If you don't see it, if you don't suppress the water. Now, I have a technique in MR. I'm making a general word of MR rather than MRI and MRS. Basically, MRI, here I mean both imaging and spectroscopy. Imaging gives the structural information. I identify the tumor, put a voxel, put a region of interest of 2 millimeter, 20 by 20 by millimeter or 8 ml voxel, and get a spectrum with water separation. It shows these peaks, N-acetyl aspartate, creatine, choline, which are now seen in the metabolic pathway that I have shown on the right side of the slide. That means now I am having a tool in the name of MRI and MR spectroscopy, which not only gives the structural information, but also the biochemical. I made a statement in the beginning of my lecture that for any cell to become a malignant or a cancer, everything has to happen at the cellular and molecular level. Here is a technique, my dear boys, that gives not only that the tumor is located inside the brain or any organ for that matter, and also gives the metabolic information or the biochemical information, is useful for me to interpret how the tumor metabolism is going to vary in a normal vis-a-vis a malignant tissue. Now, what are these brain metabolite peaks referred to? What is N-acetyl aspartate? It's an amino acid which resonates at 2.01 ppm. It normally present in high concentration in the brain cells, exclusively found in neurons and precursor cell oligocentrate. So it becomes a neuronal marker. If this peak is very high compared to the other peaks, that means the brain cells have the neurons which are viable and working all right, no problem. Suppose it reduces. The normal concentration is 10 to 12 millimolar per kg. In diseases like multiple sclerosis, encephalopathy, cerebral infarction, astrocytoma, tuberculoma, ischemia, glioblastoma, you can see the reduction. In some of them, it may be totally absent. I will give you some example, which all these would be pointed out by you by Professor KSCSTE in his talk on the neuro spectroscopy. Now, the second peak, which is the blue one that I wrote, is due to glutamate and glutamine. So this functions as an excretory neurotransmitter. That means the presence or the absence of it actually gives you an indication about the neurotransmitter status in the brain cells. And this has glutamine and GABA. So in Rye syndrome, hyperammonemia, Alzheimer, it is less, whereas in the other two, it is high. There are other diseases also which I have not pointed out because of that. Then the next big peak is the creatine, which is at 3.02. When I see the creatine peak, that means I'm going to get all the information about the energy substrate of the brain cells. Because creatine phosphate serves as a reserve for high energy phosphate in systole of muscles and neurons. So the normal concentration of creatine varies from region to region compared to N-acetyl aspartate and choline, which are more or less same in different regions of the brain. Whereas the normal concentration in white matter is 6.1, in gray matter it's 8.2, and in cerebellum it is 9.1. In the initial days of the evolution of MR spectroscopy in clinics, people thought creatine is a constant peak, and everybody used to calculate the ratio of N-acetyl aspartate to creatine, choline by creatine, etc., etc. But later there are many studies wherein it has been unequivocally proven that infectious brain masses and some tumors also have less creatine. So using this as a constant raises a lot of questions. Now the second peak is in this particular one is the blue one is the choline. Choline resonates at 3.22 ppm, present as a polar head group of lipids and membranes and myelins. That means the presence of this choline peak forming will give you the status of the brain cells as far as the membranes and the myelin. So if myelin structure is well formed in the brain, it is very important component for brain formation, and you have a big peak at 3.22 ppm. Normal concentration is 1.5 to 3, whereas in most of the tumors, it is very, very high choline. But in certain diseases, it can also be less. But in most of the tumors, choline will be very high. Then the other small peaks like myoinositol gives information again about the membrane glial cells, the membrane phospholipase, which play a role in the signal transmission. So you talk about signal transmission through the peak myoinositol. Resonate around 3.54. Normal concentration is 4 to 7. But in these diseases, it can either be low or high. Lactate, which is a peak, green peak, which is around 1.33, is normally not seen in brain cells. Under normal circumstances, a volunteer or a normal human being, the concentration being much less than 1 millimolar, you do not see it. Many people ask us the question, can I monitor, say for example, yesterday also I asked, somebody asked, can I give a drug and monitor it? No, because you are giving a paracetamol which is 500 milligrams, which may have thousands of protons compared to trillions of protons that is already there. So you are mixing 500 milligrams in a ocean, so you cannot detect it. So unless your endogenous metabolite that I have pointed out is more than about 0.2 to 0.3 millimolar in the latest 7 Tesla MRI scanner, you cannot detect it. And what I have shown is at 1.5 Tesla, naturally the concentration, if it is more than 0.5, only we can detect. So under normal circumstances, the brain cells have lactate which is less than 1 millimeter, so you don't detect. Under circumstances like in tumor, like what I have shown, astrocytoma, weight meter, cerebral infarction, ischemia, and all, you detect it. Remember one thing, as we grow from a baby to adult, the neurobiochemicals actually mature, and so as a function of the longitudinal study, 4 days, 5 months, 4 years, and adult, there is a growth of these neurometabolites over a period of time, and then sort of attain a steady state when you are adult, and all these peaks are shown here. So you need to be very cautious when you are indicating a spectrum from a children or a baby. So that's why I have shown this one. So now I'm going to give you a few examples of the brain, which I will not tell much because Dr. Keshavadas will be doing it. Just to tell you, at lactate, we don't normally see it in a normal control or a volunteer. But whereas in this particular case of astrocytoma grade 2, you can see the lactate as a reversed peak. Most of the NMR people know why it is reversed, because most of the in vivo spectroscopy, we do it at a clinical sequence with the T. So under that, depending upon the J coupling, it is inverted. See the red arrow. This is where N-acetyl aspartate, which is the best and the tallest peak in the brain spectrum, is totally absent. That means in the area that I have shown on the left of a voxel, which is true for the whole tumor, which is looking black on the left side, there is absence of neural cells. That means there is no viable neuronal cells. That means neuronal loss has taken place. Whereas the choline, which is at 3.22 ppm, is now very high compared to creatine. So this kind of information that you get. Now, in this particular slide, I have put a voxel in one corner of the tumor. But the tumor is the whole black over here on the left in the image that I am now showing it with the cursor. So what will be the neurobiochemical distribution at different areas of the tumor, as well as in comparison, whether I can also at one go compare the normal portion of the tumor. Until now, I have described about the single voxel spectroscopy. As the word implies, it is a single voxel. It's only one voxel I have put in a tumor. Now, all this experiment takes a lot of time. If we go back to this particular slide here, I now position a patient, do a global chain, which ideally you have to do it for every patient, but we don't do it. Then take MRI for about 15 minutes. Then do for another 5 to 10 minutes dynamic contrast. Then voxel position to get MR spectroscopy, the red voxel in the middle. Another 5 to 10 minutes. So 15 plus 10 plus 10, 35 minutes already over. Then I have to do some spectrum. It will take another 10 minutes. Roughly about 50 to 60 minutes. Yesterday, I made number of times the statement. One of the prerequisites for MRI is that the patient needs to be lying down in a still position without moving the organ which is being imaged. Generally, whole body has to be in a standstill position. That means for making the patient to lie inside the magnet for 45 to 50 minutes or 1 hour, like that. It's very impossible. It's a human being with life. So you cannot play around like what you do with a normal NMR sample. You put it yesterday, I think G the day before yesterday was making that 4-dimensional, 5-dimensional takes about a week or 10 days. You can't do that here. You can't do more than 40-45 minutes. So what do I do? If I have to get a spectrum from different regions of the brain tumor here in this particular slide, the black region as well as the normal one, then how do I do that? So the researchers developed the multivoxel spectroscopy. The earlier one was a single voxel spectroscopy, now multivoxel spectroscopy. Here is a case of a tumor which is white on the left panel of your slide. And then I have marked the red one from which I'm getting a spectrum. I'm showing the spectrum, but I'm analyzing the whole tumor. That means I have now divided the tumor into different small voxels, and from each one of them I can get a spectrum. I'm not going into the details of how we do that, but then there are techniques which are available and will establish. What I have seen on the right, what I have shown on the right side is the MR spectrum from the different voxels that I have highlighted in the red. Exact replica of this one. Whatever looking gray or green is a tumor. See the spectrum, the arrows blue, green. N-acetyl aspartate is less, lactate is seen, choline is high, creatine is less. Now come to the right extreme of the slide, which is a spectrum that I have got from a normal portion of the tumor, normal portion of the brain. You see now all the three major peaks, N-acetyl aspartate, creatine, choline, in whatever the ratio that they have to be in a normal tissue is present. See the differences that one can very easily qualitatively through a naked eye appreciate the spectral difference in various areas which are white in region which has the tumor cell. So this is an advantage of multivoxel. In one go, I am now having a spectrum from the whole of the tumor. See, the whole tumor is also heterogeneous. See the middle panel on the right side. There is black area inside. That means the tumor is heterogeneous. White is tumor, black is normal portion. So its heterogeneity can be talked about. The distribution of neurobiochemicals in these areas can be talked about. And then I can also compare it with the normal spectrum on the rightmost side. Now, let me leave it to Keshav Das to do more examples from his start. Now, let me shift gears and go to the other organs of the body. Now, these two examples that I have given is because that I have been doing this for the last 30 years. One is prostate cancer and another is breast cancer. I'm a gender neutral person. So I've taken one for the men's side prostate cancer and the other one for the women's side breast cancer. Nobody can find out that I am partial. Now, this is the kind of a top right is the prostate MR imaging. And what I have done basically, it is called as metabolic imaging or molecular imaging. If you want a fancy word, this is molecular imaging, what I have shown. Whatever red is the concentration of the tumor at the tumor site, and whatever yellow is the partial viable cells, normal cells is that blue over here. So this is the kind of a spectrum that you get from a normal prostate tissue, which is shown in the blue region here, which is dominated by citrate and choline by create. In the normal prostate, due to the Krebs cycle mechanism, the citrate is coming out. So you have a high concentration. Whereas in tumor site, cancer, prostate cancer, citrate will be much less and choline will be higher. I made a statement in the brain spectroscopy. Wherever, whether it is a brain tumor, a breast tumor, or a prostate tumor, any tumor of the humans, or any tumor even in the animal models, choline dominates. The reason is that choline is related to membrane structure. All of you know what is a membrane structure. When the tumor develops, the membrane ruptures. When the membrane ruptures, the choline is released. So that is why in cancer you have high choline. And this choline is basically a combination of three peaks: free choline, glycerol phosphocholine, and phosphocholine. You cannot separate them out because we are doing experiments at 1.5 Tesla and 3 Tesla, not say 16.4 Tesla or 20 Tesla. So naturally you cannot separate them out. So it's a combination of that one. So from prostate spectroscopy, what I get, the metabolic information is that I get citrate by choline plus creatine ratio, or vice versa. Or one can also determine the absolute concentration of the metabolites. I will explain to you in breast cancer, which we have. Shift your focus to the breast cancer that I have shown on the bottom of the slide. Hopefully people are not sleeping in the home when they are listening. Here, when I put a voxel in the breast cancer, what I have shown on the left bottom of the image, when I do the spectrum without water and fat suppression, then I get fat and water. As I told you earlier, breast is dominated by a lot of fat. So you get a fat peak and a water peak. When I suppress that, I get choline. Unlike in prostate, where I get citrate, choline, and creatine, in breast cancer, I see only choline. So that depends upon the organ. So what kind of parameters that I can use as a biomarker? That water to fat ratio I can do because I can determine the intensity of the area. Then total choline concentration I can do, or even I can do more. You are all now experts in the last 3 days and I can get a signal to noise ratio of the choline. This all can I use as a biomarker. So this is what I have shown, exactly the same thing. So if we put a normal breast tissue, it is dominated by fat. You see the left bottom color picture of the breast, which came in current science as a front page one. So when you do the spectroscopy of a normal breast, it is dominated by lipid, very little water, because it contains a lot of fat. So when I put the voxel, which is the red one here with the yellow voxel of a spectrum of the tumor, then you see the fat reduces, water picks up. And then when I suppress that, I get choline and some peaks of uridine phosphate, which is not consistently being observed in most of the cases. So from the normal breast, I can now get water to fat ratio because fat is there and water is there. And this is the kind of a spectrum from the normal breast. So if I now plot the water to fat ratio of, say, for example, patient undergoing therapy. Prior to therapy, in normal breast, water to fat ratio is very less. See the right one here, N28 here. So this one is much less. So when in tumor, the fat reduces and water increases. This is what I have shown on the right side. You know that tumors have different stages, different grades. So T1 is early stage, T2 is a middle stage, then T3, T4, and now involvement of nodes and other things also there. This is T4 is at advanced stage. You see the spectral profile, proton spectral profile of a breast MR spectrum. A T1 stage tumor patient, T2, T3. One can very easily appreciate how the spectral variation happens depending upon the stage of the tumor. Naturally, if you calculate the water to fat ratio, that will also change. So before therapy, that means patient has not been given any therapy, the water to fat ratio is very high. Then after therapy, it reduces drastically. That means the patients have responded to the therapy. This kind of information can easily be got. And depending upon the stage of the tumor, you have the spectral profile variation as well. Breast is very heterogeneous. under normal circumstances. So when I want to use the breast spectroscopy, like what I have shown, either from the normal area or from the tumor, I need to also keep it in mind that a woman actually undergoes hormonal changes during a 28-day cycle. What does it mean for MR spectroscopy? That means the concentration of the fat to water in a normal breast will vary. Most of the women know that prior to the menstruation, the breast becomes very soft and it becomes dense. That's all because of the variation in fat and the water that is occurring because of the hormonal changes that they undergo. So we wanted to study that, basically, how the heterogeneity of the breast is reflected in vivo spectroscopy. So that's what I have done in the left panel. We put a voxel of a normal breast, upper quadrant, lower quadrant, near the nipple region. You see the characteristic change of the spectral profile. So if you calculate the water fat ratio from the different regions, it's going to be different. So depending upon in a patient where the tumor is located, you need to cautiously interpret your spectrum. And you see, for example, then we did a study which we published in 2009. Recruited close to about 28 volunteers at different menstrual phases. It's very difficult to do. You have to convince a lady to come to the MRI during this period. And when we did that longitudinal study and monitored the 3 regions, lower and upper quadrant and near the nipple, you can see the variation, and especially near the paro region of the during the menstrual phases, you see the water to fat ratio. In the proliferative region, it is very high. Fascicular, luteal, it is less. Secretory and menstrual, it is high. So you see the variation in this one. So you need to have a cautious approach in interpreting the water fat ratio. So that's one of the limitations. Like what we did for brain spectroscopy multivoxel, you can also do the same thing. But the only difference in breast spectroscopy is that brain spectroscopy is easy to do. Also, to some extent, the prostate, whereas breast is very difficult to do it because it's an outside the human body, it's a hanging one and subjected to chest wall motion and respiratory motion. So the emotional artifact will be there, but you can still do it if you are getting your sequences to take care of the respiratory motion. It's an example of a multivoxel on the left side, big tumor of a breast cancer, multivoxel. You can see that in each of these voxel inside the tumor, wherever the tumor is, choline peak is seen. And this is what I showed as a fancy word, molecular imaging. We do the color map, and you can see the concentration of choline in the red areas much higher. And you can see the spectral profile being different. So you can also calculate the choline in the absence of absolute concentration of choline. You can also determine the signal-to-noise ratio. It is very high prior to therapy, and after therapy, it reduces. So serves as a biomarker. Shift gear to the prostate. Prostate is of a walnut size. And the various prostate anatomical zones are given here. Peripheral zone, transition zone, central gland, and all. And most of the cancer happens in the peripheral zone, which is denoted at PAZ. So this is one of the major cancers in men. Having said that, I can also make a statement that this prostate cancer is called as an indolent cancer, indolent tumor. Indolent means it is a very, very slow growing tumor. That means a patient, if diagnosed today, can live very happily for another 15 to 20 years without any problem. But that is not the case with breast cancer. Breast cancer is more virulent now, like COVID-2. Very virulent. A patient, if not diagnosed at the proper time, even a couple of weeks may be very fatal. So just bear it in mind. So if you do a normal prostate spectroscopy, you can see high citrate, less creatine, and less choline. And this is the in vitro tissues taken out during the surgery from a normal high citrate, which is now in vivo. This is right side panel is the in vitro recorded at 11.7 Tesla. Whereas it is 1 Tesla, but you still see the doublet also. This is bottom one is a tumor one. You can see the citrate being absent, choline increases. So also in the tissue that was taken out of the patient, the citrate is less. So you can do the same thing like what I have shown over here and find out what is the difference between normal, benign prostate cancer, prostate rather, not cancer, and cancer. You can see the difference in the spectral profile. You can see BPH still shows citrate as equivalent to more or less normal, whereas in cancer patient, the top one is completely absent. Choline, which was less in cancer, which is very high. And what is on the right side is the metabolic imaging, which I have already discussed. So the people at UCSF, John Kurnovics, my close friend, they made a scoring system. If you see the tablet and less creatine choline, then it is definitely healthy. Then if the spectral changes are shown as over here, then 2 is probably healthy. 3 is unequal, where equal small reduction and increase in choline. Probably cancer when choline is higher, but still citrate is in, and definitely cancer if only choline is there and citrate is not there. You can also use MR spectroscopy. I made a statement in the initial lecture that you can guide the biopsy. This is what we have done. One of my students did this. You determine the Z coordinate from the sagittal image. This is from where I took a spectrum, which is I have shown here. And then I can calculate the X coordinate. And then my student goes to the urology department and they have a biplanar test-guided biopsy. And then they feed in the coordinates and take the biopsy out. Once they take the biopsy and give it to histopathology, it will confirm the cancer. But we wanted to be sure whether the tissue that we took out from the blue square on the right panel of my slide is indeed from a cancer. So we asked a couple of patients to come to MRI after the test-guided biopsy. And when we did the multivoxel MR spectroscopy, we got a spectrum after biopsy. It still showed high choline and less citrate. That means indeed the biopsy was taken from the blue square that I have shown. Now, I will take another 5 minutes. The assessment of tumor response, like MRI and MRS, you can do that. One quick example of the breast cancer, just by using water and fat ratio, left panel top corner, pre-therapy, high water and fat ratio, post-therapy much reduced. And this is what I have shown in the values down. If you do a multivoxel spectroscopy, also you can do the same thing. You see on the right top corner, the pre-therapy A1 of a big tumor, you see the choline. And after therapy, you see the reduction in the choline as well as the tumor size. When you put a voxel in the residual tumor, choline is totally absent. Right bottom is a non-responder. That means even after third cycle of chemotherapy, the patient has not responded. So you can see the choline present even prior to therapy and after therapy, and the size also not reduced as in the cancer. And another thing is that you can also determine the concentration of the choline. This is very difficult to do, but then one of the students did it. So the concentration prior to therapy in most of the tumors in breast is 3.78, around that region, 3 to 6. And after therapy, you can see the complete reduction, and also the peak is much less. You can see on the left panel, and the right panel is from a non-responder. Before therapy, it was 4.73, top right top, and then the middle one, 4.29. It has not reduced much. And so also you can do in a number of patients and find out between responders and non-responders. You can very easily appreciate that in responders, 4.8 millimolar per kg was the concentration reduced to 0.4. Whereas in non-responder, the 2.1 millimolar concentration that we noticed in most of the tumors stayed more or less the same even after third cycle of chemotherapy. This is one example, not from my work, from John Kurnovics' work. Successful therapy of the metabolic atrophy. What you get basically is metabolic atrophy. So this is prior to therapy. After therapy, you can see most of the peaks have disappeared. That means no viable cancer cells to give the citrate peak of the choline or the creatine peak. That's why right bottom, you see the complete absence of the after 34 weeks of androgen deprivation therapy, which is called as metabolic atrophy. This is another example where therapeutic failure is also there. That means it is not successful therapy. You can see that even after the therapy, you can see the choline and creatine peak in this one. So it is not only used for assessment of tumor response, non-responders also, where even the therapy fails because of the newer drug, then you can also do that one. You are not only confined to doing proton. You can also do other nuclei, but it's very difficult. So one such example that I will give you, just a flavor, it's only for the sake of completeness so that you can appreciate, because most of you are physics, chemistry, and biology, appreciate that other nucleus can also be done. So this is the kind of a spectroscopy which is from lithium ion. Remember one thing, lithium drug is given to manic depressive patients. So we constructed, when I was doing postdoc, this is my boss, Professor Richard, from whom I learned the technique of in vivo MR. We built a lithium coil, and whatever lithium that you give it to the patient only can be detected. The reason is that this drug acts as a tracer material. The normal concentration of the lithium in the brain cannot be detected. So also sodium and other things. Okay, sodium, we don't do that, but I'm telling you. So any drug that is given to many depressive patients is basically lithium carbonate in divided doses or in one dose of 900 milligrams per a day. So we wanted to determine how much of concentration of the lithium goes into the brain. In psychiatric or manic depressive patients or maniac patients in general, the choice of the drug is lithium. What should be the seat of action? It should be the brain, because most of the psychiatric disorder comes because of the ionic imbalance. Lithium is an ion. So we wanted to find out how much of the lithium carbonate drug that is given goes into the brain. That is very important. It will go all over the body, doesn't matter. But what it goes to the brain is very important for me. Can I have a technique by which we can determine? There are only one technique which you can do it is flame photometry. But that gives the information in the blood. What you get in a blood is not going to be the same in the brain. So we want to determine the lithium concentration in the brain. So we built a lithium coil, and we can very easily tune to lithium, which is very close to that. So, and then we took a standard and do that one. So we did an experiment of Vietnam veterans. Vietnam army men who were there for so many years, they have many depressive illness. So what we have done on the right side is the brain concentration. And we also did in the serum, because that goes everywhere. And we also determine serum level. One is cough, one extremity of the human body, the other extremity is the head. So we find out that the concentration is in brain, goes, and then dips after 7 days. So then we found out actually why it dips, whereas in the serum also it dips, but in cough muscle it doesn't decrease. Reason is that this particular patient was non-compliance of the drug, because as I told you, whatever the lithium that you detect is coming from the drug that you are giving, and it is getting concentrated in the brain cells. So it acts like a tracer element. So normal concentration that is present in the normal human being, you cannot detect. That's why if we are non-compliance of the drug regimen, that also can easily be found out, and we determine the concentration. Another study we did is, this is also a dynamic MR, functional MR, is that 5-fluorouracil is normally given for most of the cancer, like breast cancer and all. And these 5-fluorouracil chemotherapy agent is given a modulator like methyltrixoide, MTX, that I have shown in the middle and the right panel. So the reason is that whether it increased the absorption or not. So we wanted to find out. So what we did is that we again built a fluorine coil. As all of you know, when you take a drug, say like even crocin, paracetamol, what happens? It goes to the stomach and then liver metabolizes. Liver is the seat of action. So we put a fluorine coil in the liver, where the anabolism and catabolism takes place, and then we monitor it as a function of time. So as and when we give the injection to the patient, this 5-fluorouracil with this one, then we take the spectrum. Again, like lithium, fluorine in human body present cannot be detected because it's a low concentration. So what you detect is from the exogenous agent, which is given as a 5-fluorouracil, period. No more question on that. So the moment at 3.5, you see the fluorine peak. And as soon as the drug reaches the liver, it metabolizes, and then in catabolic pathway, you see 5-beta alanine. Fluoro-beta alanine is coming out in higher quantity. So that is what you see, for example, at 10.5 minutes. The middle one is where we get the modulator before 5-FU, and here after 5-FU. So there is no absolutely difference. That means giving the modulator before or after does not make a difference. Not a very great information, but still information. In clinics, even if a drug doesn't work, it's a positive lesson because you know that it doesn't work. Now, what is the future of in vivo spectroscopy? You see what I have shown on the left panel, 1.5 brain spectrum, 1.5 Tesla, 3 Tesla, middle one. See the number of metabolites slowly start increasing. At 7 Tesla, you see a rich group of metabolites biochemicals that you track. It means in addition to N-acetyl aspartate, creatine, choline, myoinositol, I have now glutamate, glutamine, aspartate, so many, you name it, you have it. So you now have so many biochemicals to interpret your metabolic pathway. So also is the case of breast spectroscopy. What I have shown on the right, 1.5 and see at 4 Tesla, so many peaks are seen. In 1.5, we saw only total choline. Now you are seeing tarbine, glycine, alanine, which can be correlated with the in vitro tissues as well. So the take-home message is that MR is a powerful technique wherein you can get an image from any organ of the body, from any extremities, and then not only that, you get the morphology, you get the complete physiology and metabolism. This is the only technique which was developed by physicists, later hijacked by chemists, biology, and then to medicine. All are joined by a ladder called nuclear magnetic resonance. Growth is so much enormous over a period of time that it has sort of revolutionized the clinical medicine in total. It's the slide that I borrowed from Richard Hans. So I would conclude by thanking most of the work that I have given today and some of the work yesterday are all from my own group for the last 3 decades in AIMS when I was there. So many people, some of them I have omitted here, but most of them are here and my PhD students and postdocs over the period of time. And thank you very much for your work.
Thank you, sir. That was a wonderful talk, especially because you told how it comes from physics, biology, going to medicine. The application part you have highlighted extremely well. And therefore, you have made my job also simpler because I will be looking from more the application side.
Yeah.
Because my job is actually interpreting these images, interpreting these signals. So I will be focusing on that. Any questions?
There are few questions. Sir, which technique is more efficient in learning bone tumor or cartilage-related issues? X-ray scans or MRI? Maybe Keshav, you can take it.
Yes, actually, in my talk, I will be telling about the answer to that. For bones, the question is for bones, which is useful, isn't it?
Bone and cartilage. Cartilage is MRI better. For bones, it is X-ray, which is
exactly. So articular cartilage for osteoarthritis, for example, the MRI has a specific role, but the bones always it is the CT scan which is useful.
The second question is, if MRI is performed, can an MRI be performed on a pregnant woman? Then is it dangerous to the fetus?
Yeah, so usually we try to avoid it in the first 3 months.
First trimester.
Because that is the time when the organs are forming, organogenesis is happening. So at that point, we do not. Also, we avoid this contrast media also as much as possible during the pregnant for the pregnant woman, because we do not know the long-term effects. There are not much studies on pregnant females of injecting these contrast agents. So it is better avoided in the first 3 months. Afterwards, if it is required, you can do it. There should be no problem.
I will just add to what Professor Keshav Das is telling. Okay, I gave these questions to Keshav Das is that he's a practicing radiologist. You know much more clinical side than me. Well, just to add to this one, basically in most of the clinical medicine, if the advantage of the patient is going to be more by doing an MRI, it will be done.
Yes.
If it can be satisfied with the CT scan or other method like ultrasound, they will do it. This is the sort of an unsaid general rule. Am I correct?
Yes, it is called the risk versus benefit ratio.
Benefit.
Any surgery is being done. If there is risk more, we avoid it. We tell the risks are more, benefit is less. So this ratio is looked into before treatment of any disease.
Yeah. The other question is, can MRI help to detect upcoming event in the brain? A very interesting question. See, one thing I will say, I will leave it to Keshav, but one thing before he answers, I will say that MRI is not a screening modality. Keshav, you can fill up.
So I would put like this, for example, and I will show you some one case also. Sometimes, some things are not seen in MRI, but this MR spectroscopy or some of these advanced MRI techniques might give us some biomarker that some change is happening in the early stage pickup. For example, Alzheimer's disease. You might have all heard about Alzheimer's disease. Before Alzheimer's disease, there is this entity called mild cognitive impairment. The patient who is above 55, 60 years slowly starts having memory problems. That is mild cognitive impairment. At that time, you start looking at the MR and start seeing it. Then later on, when you see all the main changes of a volume loss of the brain, temporal lobe, hippocampus, and all are seen. But the earlier stages, so this is possible. But even in the early stages, you can pick up some of the diseases, like, for example, Alzheimer's disease.
One more thing I will add, which we ourselves has done, alcoholism. So before the brain damage, as if you do it on alcoholism, you can find the brain metabolite changes. But the only thing is, if you know the patient has Alzheimer, a little bit early symptoms, you subject them. But you cannot put anybody and say that there is going to be an evidence for that. It's just, yes. Great talk. What is the workaround for the subject's movement during multivoxel scans? See, basically, we do the multivoxel also in 5 to 6 minutes along with the imaging and other things. Total 40-45 minutes, most of them do it. But nowadays, if you do only MRI, non-cooperative patients, we have a lot of sequences which can be done in them. Even with a slight 15-degree movement of head, we can do that. Spectroscopy cannot be done with that one. So MRI can be done with non-cooperative patients. There are sequences in which even small movement can be taken care of.
Any other questions? So, sir, thank you very much, sir. It was wonderful hearing you. We learned a lot, actually. I'm sure that students here would have learned the applications of this wonderful technology. Thank you, sir.
Yeah.
Thank you very much for this talk. Wonderful talk.
Yeah, thanks, Sunil. Yeah.
Thank you, sir. Thank you, sir.
Yeah.
So we will go to the next talk.
Yeah. Paulson sir will introduce you first, then he is sharing the session.
Yeah.
He is here.
Good morning, all. Now we have already Professor Keshav is with us. The Professor of Radiology, Prerequisite Institute for Medical Sciences and Technology, Trivandrum. He is an expert in various aspects of various branches of neuroradiology. For example, MRI, pediatric neuroradiology, epilepsy, and he got epilepsy imaging. He got training from various international institutes, for example, Department of Neuroradiology, Scientific Institute and University Hospital San Raffaele, Milan, Italy, in MRI neuroradiology. And Department of Pediatric Neuroradiology Hospital for Sick Children, Toronto, Canada. And in the case of epilepsy imaging, Department of Clinical and Experimental Epilepsy Institute of Neurology, Queen Square, London, etc. He is interested in magnetic resonance imaging, neuroradiology, medical imaging, processing. He has already published for nearly 250 papers or articles in international peer-reviewed journals, having an H-index 35, and he got more than 5,000 citations for his articles. He's a editorial board member of various radiology-related journals. He received various international national awards, for example, National Bioscience Award in 2009, award by the Department of Biotechnology, Government of India, for the work in functional neuroimaging. Dr. Ashok Mukherjee Memorial Award in 2006, award at the annual conference of Indian Radiological and Imaging Association, Chennai, and Cum Laude awarded by the Radiological Society of North America, Chicago, USA, for the scientific exhibition. These are some of the awards, and he's a fellow or member of the various academic bodies. For example, President of Indian Society of Neuroradiology, President, Indian Chapter of International Society of Magnetic Resonance in Medicine, Fellow of the Indian College of Radiology and Imaging, Member National Academy of Medical Sciences India, Member National Academy of Sciences India, etc. And today he is going to deliver magnetic resonance spectroscopy for studying brain disease. And his talk involves two parts. One is the basis of image formation in MR imaging, and second one is the MR spectroscopy for disease diagnosis. We all know that this MR imaging is a very popular technique now and widely used by by doctors or physicians in diagnosis, especially in brain tumor or epilepsy or brain-related disorder identification, etc. And on behalf of the organizing committee, I invite Professor Keshav to deliver the talk.
Thank you, sir. I'm just trying to share my screen. Let me see.
Okay, it is. Yes, it's not visible now.
Yeah, you presented it. You minimize your.
No,
yeah,
can
it's
you
coming.
see?
It's coming. It's coming. Yeah.
It's okay.
Now you can see?
Yes.
Okay, fine. So, thank you, Magnetic Resonance Society, as well as the Srinivasa Ramanujan Institute of Basic Sciences for this invitation. So, I started doing MRI in 1994. In 1983, when I actually joined for medicine, at that time there was nothing called MR and CT in our institution. I studied in Calicut Medical College. We did not have any of these imaging techniques. So from 85 onwards to 94, I did not know what MR is and how it will be useful. Most of our diagnosis that we used to do of neurological conditions or cardiac conditions, everything was based on the tools that was available there. Auscultation, that means with a stethoscope we used to try to find out or look at the clinical symptoms of the patients. From that, now as you know, the whole field has become highly, what do you say, machine dependent. And one of the techniques which has really revolutionized the way we look at the human body is MRI. And MR spectroscopy be just one technique in MRI. So the learning objectives of this talk will be a short introduction to the MR sequences. Then we will go to the MR safety, the basics of MR spectroscopy, which was discussed by Dr. Jagannathan. Then I will go to MR spectroscopy of the brain diseases. Then discuss some things about the other techniques like perfusion, diffusion, tensor imaging, and all these things, and give a broad idea about MRI imaging biomarkers. So as you know that in one of the last slides which Professor Jagannathan showed, he showed how physics and chemistry, how these basic sciences has influenced biology and later on medicine. And it is at the level of applications that human beings understand the importance of basic sciences. Yesterday in our convocation, we had the chief guest as Dr. Soumya Swaminathan, who is the WHO scientist. And she was telling how genetics has influenced, genetics and biotechnology has influenced the treatment of diseases. Gene editing and all these techniques have come, and many diseases, which congenital diseases, have been treated. So there, the biology has played a role. Here it is the chemistry and physics which really made such a change in the way that you look at human body. The only thing is that the MR for medicine came much later. Lutherberg and Mansfield got this award only in 2003, while the basic principles of NMR happened much earlier. The physics and chemistry awards were given much earlier. But at that point, just like any controversy in awards is there was a controversy in the award in which MRI got for application, medical application, and that was in US. At that time, Professor Dr. Damadian had come out with an MR in which he had already started doing some human parts. MR was being done. His MR is called the Phonar. So they were already doing it. And his students were quite surprised when the award was announced and his name was not there. They expected at least his name would be there among the recipients of the MRI application in medicine. But that did not happen. So the United States, of course, recognized his contribution, and the National Medal of Technology was given to Damadian and Lutherberg. Lutherberg, of course, was given the Nobel Prize as well for MR. So actually, Damadian was a physician, a medical person, and he started the applications in medicine. So from there onwards, there has been a lot of change which has happened. And this is the first MR that we have. I was telling you about the MR in Sri Chitra Institute, which came in 93, 94. It was at that time which came. This is our first MR. It was a GE magnet. And now we have another two MRs, which is a 1.5 Tesla machine and a 3 Tesla machine. This machine is now not being used. It is gone, the old GE machine. And the person who does the MR is the MR technologist. The MR radiologist is the person who interprets the images. The technologist knows about the sequences which has to be done, the protocols which has to be followed for a particular disease. So say you are doing a patient with stroke, there is a particular protocol. There are certain sequences. And I'll explain to you what are these sequences that we usually do. And similarly, you have this for stroke, there is a protocol. For dementia, person coming with memory loss, there is a protocol. So there is protocols for all these things, and we do according to the particular disease. And towards the end of the imaging, the radiologist sees the images and says that, okay, I don't think you have to do anything more. This is enough. Or if you have to add any further sequences, it is decided at that point of time. In some cases, for example, the contrast media has to be given. These decisions are made. So usually in the console, you have the technologist sitting, the radiologist also will be there. And they have to look at the images at the same time, look at the patient, the safety of the patient has to be looked into. All these things has to be considered by both the technologists and the radiologists. Now, I will just go in short to discuss about the MR physics, because you need to understand what are the sequences and what sequences we actually work on to get a useful image. So you know that before a patient is put into the magnet, the hydrogen nucleus, which is having a single proton, moves in different directions in this fashion. So our body is full of these hydrogen nucleus. So this is what is usually used for imaging in MRI. But when you put it in the magnet, many of these protons are aligned in the direction or on the opposite direction. Most of them in the direction aligned parallel to the magnetic field. And then it is not just the aligning, there is also spin which is happening. So protons just don't align, they go having a spin, and that has a particular frequency. So they also spin at a particular frequency. That is called precessing. So these two things are happening, and as a result of which there is a vector which is created, mostly longitudinal. A longitudinal vector is created, which is then flipped by means of a radio frequency pulse, either 90 degrees or 180 degrees. And when it is flipped, and then when the RF pulse is removed, then the echo is created, and it is the echo which is picked up by the magnet. So many of these things you might be knowing. I am telling from a clinical viewpoint to make you understand certain particular words which are very important, like longitudinal magnetization and transverse magnetization. So at this stage, it is longitudinal magnetization which is very high. There is hardly any transverse magnetization. But after the RF pulse is applied at 90 degrees, you can see that longitudinal magnetization goes down and the transverse magnetization increases. So both these phenomena, the vector which was pointing, the longitudinal is flipped, and this precessing also, there is a change which happens when the RF pulse is removed. So the longitudinal magnetization again starts growing. So why this is important? Because the time taken for the magnetic vector to return to its resting state. So resting state is the longitudinal stage, gives us an information about this time is called the T1. And the T1 varies according to the tissue. So you have a particular value in the brain. So if you, in the brain, as you know, there is structure called the gray matter. Gray matter is where most of the neurons are there. That is the gray matter. Then there is the white matter. And then there is the cerebrospinal fluid. There is a lot of fluid within the brain. And there is fat all around, especially in the scalp, in the skull and all, there is a lot of fat. So all these structures have a different value of T1. So the relaxation which happens, the time which is called the longitudinal magnetization, the time taken for that, as well as the time taken for the T2 to come back. So that means the transverse magnetization to come down is called the T2. So these two time constants are what we are looking for. And again, the T2 is different for the gray matter, the white matter, and this cerebrospinal fluid. So basically, we are looking at the T1 value and the T2 value, which are time constants for various tissues in the body. And this varies between gray matter, white matter, cerebrospinal fluid. Okay, so you understand about the T1 and T2 now. You understand about the transverse magnetization and the longitudinal magnetization. How the longitudinal magnetization first decreases when the RF pulse is applied, and when the RF pulse is removed, the longitudinal magnetization grows back again, emitting an echo. Now the echo is released. Now from where is this echo coming? That we have to find out. For that, we apply some other magnets, magnetic gradients, and that gradients are called the slice selecting gradient, the frequency encoding gradient, and the phase encoding gradient. Three gradients are applied in XYZ direction. And from there, we know that from where this signal is coming from the body, whether it is, so suppose you are looking at the brain, whether it is coming from a structure called hypothalamus, or whether it is coming from pituitary. All these things is decided by putting these various gradients. So now you know from where the signal is coming. You know the T1, T2 based on the transverse and longitudinal magnetization. And then we go to the most important thing, and that is the sequences. And what the radiologist will be playing with is these sequences. So Jagannathan sir has already told you about the free induction decay, which is used for very commonly for MR spectroscopy, where one RF pulse is applied and you get the FID signal, which you would be knowing very well. Then there's a gradient echo is there, there is a spin echo is there, stimulated echo based on the RF pulses. But it is the RF pulses when you apply and when you get the echo which matters the most. So for example, in a simple spin echo sequence, that is the most common sequence that we do for brain. In the brain, we do a spin echo sequence, and in the spin echo sequence, we first give a 90-degree pulse, which flips the longitudinal magnetization 90 degrees to the transverse magnetization, and then we give a 180-degree pulse, which flips it 180 degree, and after that we get the echo. So this is a spin echo. This the first one, that signal that we get is the FID, and here you get the spin echo. And this spin echo is, as you know, dependent on two things. One is the TR that you apply, time to repeat between one 90-degree pulse and the next 90-degree pulse is the time to repeat, and the time to echo when you get the echo. So these are the two important parameters that we have to play with to get a particular sort of image. So this is how you draw a sequence, and if you have to draw, you have to tell what are the pulses, that RF pulses that you apply, a 90-degree pulse, a 180-degree pulse, then the size, where you put the size slice selection gradient, the phase encoding gradient, the readout gradient, the three gradients to look at the position from where the signal has to come, the analog digital converter from where at what point of time, and finally the signal being obtained. So every sequence in that way has a particular drawing, and it is based on, so whenever an MR physicist looks at it, he understands that when is the RF pulse applied, when is the slice selection gradient, when are the signals are being obtained. So it is a spin echo sequence, as I told you, is one of the most common sequences that we do. We have other sequences like the gradient sequence, and some of the sequences are gradient sequences. Some are now echoplanar sequence. Why the echoplanar came? Because we wanted MR to be faster, because there was a question that, you see, some of these patients do not lie down for a very long time in MR. So we need to make the MR technique short. For example, a patient is coming with stroke. This stroke patient is not going to lie down in the MR for 10-15 minutes, even 10-15 minutes. They may lie down for some 2, 3, 4 minutes, and in that 2 to 3 or 4 minutes, we have to get all the information. So in that case, can we make sequences which are short rather than this applying this 180-degree pulse? Can you just do with your 90-degree pulse? So there are techniques like the gradient echo techniques are there. Then there is the echoplanar imaging technique is there. Faster imaging techniques are available. So, and there is a lot of work today happening by MR physicists to make the whole technique of MR faster, even to the point that artificial intelligence is being used. Machine learning techniques are being used along with these sequences. Image processing techniques are also being used. Machine learning techniques are also being used to make MR faster, because still the radiologist thinks that the time taken for MR is quite high. Then I was talking about TR and TE, time to repeat the pulse, 180-degree pulse, time to echo when you capture the echo. Based on your value of TR and TE, you get images which are T1 weighted, T2 weighted, or proton density weighted image. When you put a long TR and a long TE, then you get a T2 weighted image. But if it is short, the TR, the time to repeat and the time to echo are short, then you get the T1 weighted image. And here it is long versus short. So this sort of a combination of timing is decided by the technologists. In most of the machines today, whether it is Siemens, GE, or Philips, already these are well written and the protocol and the sequences are well made there. And you have to just switch on the system and then go to these parameters. The parameters are all selected and kept. And whenever you want a T1 weighted spin echo sequence, you just press buttons and you get it. But the technology also knows and has to know, as well as the radiologist has to know, that it is based on these TRT parameters that actually an image contrast comes. It becomes a T1 weighting or a T2 weighting. Because sometimes when there are mistakes which happen, artifact starts coming, then sometimes this TR and the T values are responsible. You would have given a very long TR and you would be, or a very long T, and you would be expecting some T2 weighting in the MR image, which will not happen. So this is again an important knowledge that you need to understand. So this is, for example, these images which were shown very nicely by Jagannathan sir. So these images are of the brain. You can see that what I was telling you, this white structure is the cerebrospinal fluid, the fluid within the brain. All these white structures are all fluid. And here there are two types of tissues you can see. This thing in the periphery here is the gray matter. And towards the center is the white matter. So this looks gray in T1 weighted image, and this looks more white in the T1 weighted image. So that is the white matter. So whatever you see in the T1 weighted image, but it is almost slightly different in the T2 weighted image. The white matter appears more black, and the gray matter appears differently. And here you see in the proton density images, all the blood vessels in the brain show up very nicely. So every sequence and every method, this is the spin echo sequence, and every weighted image, T1, T2, and proton density image or the flare image, there are many methods like that, gives a particular information. For example, the vessels are not, as you can see, not that well visualized in the T1 and the T2, but it is good in proton density. So this is an information which the radiologists know, and that helps in the interpretation of the image. And another thing which is important is, in any plane you can get the images, which in CT scan initially it was very difficult. You only used to get the transverse image, the axial image you call. Now you have the coronal image, the sagittal image, and you can even get it in different planes if you want, based on how you plan it in the brain. So you have a particular plane, two structures are taken, it's called the anterior commissure, posterior commissure. You plan along it. This is the universal standard that we use, and the cuts above and below are looked into. But when you are looking at a patient with epilepsy, sometimes you will have to plan along a structure called hippocampus. Now these are the informations which the radiologist and the technologist knows. And while interpreting the images, we have to look into these things. So these are in different planes. Similarly for the spine also, by now you will be telling, this is a T1 weighted image. Why this is a T1 weighted image? Because CSF is black. The CSF, the brain and the spinal cord is within a fluid, and that fluid is called the cerebrospinal fluid, and that is black in the T1 weighted image. In the T2 weighted image, it will appear bright. But there are other structures. The fat you see, this is the fat in the back of the neck. There is so much of fat. And that fat is very bright in the T1. It is not as bright, but in T2 also it can be bright. So you look at the bone marrow. Here the bone marrow is there, and bone marrow has a particular signal. When these signals changes, then that means there is some abnormality which is happening. So we have to first know what is normal anatomy, and then go to the abnormal. So the most important thing, yesterday Dr. Rajendran was stressing this particular point, that tissue characterization is the most important thing which MRI and CT and ultrasound and all these imaging techniques do. We have to, the liver will look different from spleen, might look different from some other structure in the brain. Similarly in the brain, the gray matter looks different to the white matter which you have already seen. The fat looks different. Blood, hematoma is blood in different stages. Early blood looks in one way, late blood looks in another way. So this hyper and hyper, which is written here, denotes the signal. So what is hyper? Hyper is brighter. It is more bright. Hypo means it is more darker. And iso means it is neither bright nor dark. So looking at brightness in the signals that is obtained in the MRI, we can tell the tissue. But example is water. Water is usually, as I have already shown you, the cerebrospinal fluid. It is black in T1. It is bright in T2. It is almost ISO in CT scan. So based on these things, everything has that sort of an appearance which helps us to tell what tissue it is. Now this is the question that somebody from the audience was asking. How it compares, MR compares with CT scan. We never think that CT is in any way inferior or ultrasound is inferior, any structure like that. For us, every of these imaging techniques are complementary. Each has its own advantage. The advantage of MRI is that soft tissue visualization, brain structure visualization like the white matter and all are much better seen in MRI. But look at the number of plus when it comes to bone, calcification, calcium in the brain, or because of the previous surgery, some metals are kept inside and all. These might be sometimes missed on MR. Sometimes some of the material which does not cause an artifact might be missed on an MR, are picked up very nicely on the CT scan. So based on a particular condition, we decide that this patient should go for an MRI or a CT scan. Having said that, I just wanted to tell there is one new technique which is coming in, which is called the ZTE or 0T. I was telling about the TE. So T is the time to echo. A new sequence is coming up, which is called the 0TE, which has a wide application in orthopedics to look at the bones. And all this is coming. This is a new sequence, and we had a paper this time in the International Society, which happened last week. It was an online meeting, but it was a hybrid meeting. The other was in Vancouver, and our paper was on ZTE in the base of skull, because skull has a lot of bones, and sometimes you have to do both the MRI and after that the CT scan to evaluate the skull bones. But what we were telling is, if you use this technique called ZTE, you will be able to pick up even bony changes. So MR is evolving. From 94 onwards, when I started seeing MR to now, the number of sequences have increased so much that many of the things which were not possible in 94 are now being made possible because of the technology. And this is mainly because of the development in MR physics, especially the sequences. Now, this was about the contrast which Jagannathan told yesterday. We use gadolinium. Gadolinium is not injected directly. It is chelated with an agent like EDTA or some other agent by means of which it is made much safer. Otherwise, it is toxic. You cannot just inject this heavy metal there. You made it safe and then inject it. The only problem of injecting gadolinium is in actually patients with very severe renal failure. In those patients, there is a problem of a condition called nephrogenic systemic fibrosis coming. Nowadays, this condition is decreasing because people have recognized it. So they don't inject in such patients. But in most of the other people, you can inject. And this question was also there, whether in pregnant females. So usually we try to avoid it. And then we look at the risk to benefit, and then only we inject it. Now, I was talking about this common 2D sequences. The sequences which we usually do are the spin echo sequence, and which gives an image of the T1 weighted, the T2 weighted image. The T1 weighted image gives a good information about the anatomy, while the FLAIR is the sequence, fluid attenuated inversion recovery sequence. This is a sequence which gives an information of pathology, almost always done. And the conditions like multiple sclerosis and tumors and all, it gives important information. Proton density, I showed you it shows the vessels and all, but nowadays it is much less done, proton density. Then you have the gradient sequence, T2 star, which is very good in picking up blood and calcification and all. Of course, not as good as CT scan, but it is a very good sequence. Modification of that is the susceptibility weighted sequence that we use. Diffusion weighted image, at least some of you in the audience might be remembering the importance of this, what Dr. Jagannathan told in stroke detection. Because you are looking at the diffusion of water molecules, and in stroke there is a restriction. The water molecules are not able to move around in the brain, and therefore that is picked up by the diffusion weighted sequence. STIR is a sequence used for fat suppression, and we don't want in the certain areas in the body there is a lot of fat, and the fat will obscure the pathology, the disease. So for removing the fat, we can use the STIR sequence. Then there is the inversion recovery sequence. These are the various sequences which are done 2D, and there are also sequences which are done on 3D. 3D means you get more amount. If it in the 2D you have about 35 slices of the brain. In 3D you might have about 140 slices. No gap between slices, very thin slices, so that you get very good information. These are some of these sequences: 3D cis, flare, SSFP, spoil gradient, and all. Now this is an example. For example, this particular, there is a tumor here. You can see a tumor here in the brain, and this is a 2D sequence. Now here you are not actually sure whether it is inside the brain or outside the brain. I'm sure people from physics and chemistry and biology might find it slightly difficult. This is interpretation. This is what is my job. So I am just telling you. But if you use the 3D, you get much more thinner sections, and then you can once you get the 3D, you can also look it in the axial. You can do reconstruction in the machine itself to make it as axial. Once you acquire it in axial, you can make it in coronal, sagittal, and then actually look at the attachments of this lesion from where it is arising is much better visualized. So by means of a 3D sequence, or it's also called Fiesta C, you are able to detect that this lesion is outside the brain stem. It's not inside. So this information you get by these 3D techniques. I just showed you an example, but 3D sequences are becoming more popular now. It takes more time, but because people are working to reduce time, we can get it in 6 to 7 minutes, while the 2D sequences might take about 2 minutes or maximum 3 minutes. This is the diffusion weighted image, which picks up stroke. So these are all sequences which are very useful. And the next thing is the quality of the image. So you cannot just take any quality and start interpreting these images. The image has to have a particular quality. And what are the factors which improve the quality of an MR image? That is the spatial resolution, the signal to noise ratio, and the image contrast. I have already talked about the image contrast, the T1 weighting, the T2 weighting, the flare weighting, and all these things. So it is based on parameters like TR, T, and the flip angle. Flip angle is from 90 degree, longitudinal magnetization to transverse magnetization 90 degree, or you can even flip it much lesser, 30 degree or 60 degree, and that is what is the flip angle. Number of acquisitions that you give, the field of view, the matrix size, the bandwidth. These are all things which matters when you actually look at the image quality. It is just like when you take an image using your camera. There are a lot of focus point and the brightness and all these things you look before you take the photograph. Similarly, in MR, there are a lot of parameters that you have to look into before you take the image. Example is SNR, for example, signal to noise ratio. If you do an MRI on a 3 Tesla MRI machine versus a 1.5 Tesla MRI machine or the older 0.5 Tesla MRI machine, the SNR will be definitely better on a 3 Tesla MRI machine. Similarly, spatial resolution can be changed if you change the matrix size or the FOV. So there is these things can be changed to get a better quality image. Next, we go to the safety issues of MRI. And this was touched upon yesterday, but I will give you, this is one of the most important things that the technologist and the radiologist is concerned about. So you can have biological effects due to the static magnetic field, the RF related injuries, the gradient fields you apply, the 3 gradients that you apply, and the acoustic noise. Some of you at least would have gone into the MR machine, and especially if you have gone into a 3 Tesla MRI machine with a very high gradient strength, the peak amplitude and the slew rate, if it is very high, these are some of the values which looks at how fast the gradients grow. If you go to these machines, the sound will be so much inside that some people cannot lie down in that much sound. So you have to give them plugs in the ear. Then these operational hazards, this is very important. Many hospitals have these problems, and I can give you some example, especially the ferromagnetic objects all around you. There are so many ferromagnetic objects in a hospital which may be sucked into the MR. Then implants, patients with implants have to be very, very careful, which are especially susceptible. Like, for example, there is a very old implant which has been placed with this made totally of iron and all. That person cannot go inside the magnet. Nowadays, most of the implants are MR compatible. They are making it MR compatible. Then this projectile phenomenon, which even recently happened. So what are these problems of field effects? Field effects are the 1.5 Tesla magnet or the 3 Tesla magnet, and the field effect because of that. People with particular surgical clips cannot go inside. Prosthetic valves, these are all the older prosthetic valves, the older surgical clips and all were not MR compatible. These are ones which are put in 80s and 90s and all. May not be MR compatible. So MR patients cannot go. This mascara is a very common thing. Actually, we always tell females who are coming that don't put this eyeliner and all these things. That might contain iron. So these patients can have burns after in the eyelids after the MR, because of this magnetic effects, because of the iron in this mascara. So these are things which has to be looked into. Very careful examination of the patient has to be done. The patient has to go through a door-like thing to see whether there are some metallic objects, just like what you do in airport, and see that if there are some metallic things like coins, watches there, keys, these are all will fly into the magnet. For example, if you go inside the magnet with a watch, the watch is going to stop. If you are going to the magnet with your this credit card, it's not going to work after you come out. So these are all actually written down outside. A board like this is kept, but still many patients do not understand these things. Even in local language, if you write, they in that tension, they may not look into all this. And then it is the responsibility of the radiologist and the technologists there to look into this safety aspect. This is the pacemaker. With the pacemaker, again, patients can have problems in that magnet. Then the RF burns. Suppose there are a lot of coils. The ECG machine is connected to the patient, and some other tubes with iron is connected, and it is looping around the patient just like this. You see, the patient is there and it is all looped around. Then there can be local burns which can be produced, pulse oximeter and all. So these are things which has to be taken care when the patient is going inside the magnet. These are some of the hazards which have happened. India as well as abroad, this has happened. Oxygen cylinders flying into the magnet. Sometimes it flies along with the person who is carrying it, actually causing major injuries to the person who is carrying it. So such major complications can happen. So the chair or sometimes even the bed on which the patient is lying, if it is not MR compatible and the patient is taken inside, they can develop all these MR hazards. So extreme care has to be taken while doing MR, and this is not for just the technologists and the radiologists, but it is also important for MR researchers. Nowadays, a lot of people from physics background, biotechnology background, or chemistry background, they are interested in MR research and coming to institutions, medical institutions, and doing it. And sometimes they don't have this understanding about MR safety. And actually speaking, in most of the countries abroad, they have to undergo a MR safety course. And that is extremely essential. And they have to sign that they have actually undergone this MR safety course before taking a patient for research into the magnet. And this is from the ISMRM, International Society of Magnetic Resonance Imaging. They have put up some criteria. You have to look at for the screening for the patient for all the things that I mentioned. Magnetic field, whether whatever the patient is having inside the body, whether is MR safe or MR conditional. You can get this information from the internet, mrsafety.com. If you go, these informations will be there. What are the materials which are safe, which are MR conditional, and which are the ones which cannot go inside. Then acoustic protection has to be given at a proper point of time. I have told you about the RF field, coiling of the cables around and all could be a problem. And then this is the most important thing, that trained and educated people, the staff who are going to work in this area. Because in a hospital setup, unlike in a research lab, you are not going to have just people who are trained. There are a lot of people untrained who are going around. There are sweepers, there are people who are bringing in this oxygen cylinder or pushing their patients. They do not go through a training process. And sometimes they are the ones sometimes can make a lot of mistakes. So it is essential that departmental or an institute MRI safety board is there, which looks into all these things. One of the most common issues, however, that we have seen is the fear of closed space, which is the claustrophobia. Many of these patients have this when they go into this 60 centimeter bore magnet. They have so much of tension. They say that we feel panic attacks. We are not able to go inside. So in such cases, nowadays, the bore of the magnet has become bigger. It is a 70 centimeter bore. And what we have seen is we have a 60 centimeter bore as well as a 70 centimeter bore. Whenever we have problems, we take these patients to the 70 centimeter bore, and many of these patients find it okay. But still there could be people who say that, no, I am not able to go inside. And these open magnets are also quite useful. Now about MR spectroscopy, Jagannathan sir has already given a very good introduction to it, how we are looking at chemical shift based on the environment in which the protons are there, whether in water as well as lipid. It will have a difference, the parts per million that you get in the x-axis, and the y-axis here, which is shown, not shown here, gives that information about the amplitude. And I will go into that in the subsequent slides while I'm describing about the basics of MR spectroscopy. So you can use either the steam technique, the stimulated echo technique, or the press technique, the spin echo pulse sequence. It is the pulse sequence which commonly is used is the press sequence. You can use the single voxel, a limited area is covered, gives a good information. But as sir was telling, the problem is a very small area alone can be covered, and you might not be able to look at the opposite side of the brain or around the tissue. Around, if there is a tumor around the tissue, also there could be abnormalities that cannot be picked up. So we have been using multivoxel spectroscopy for that, and it has advantages. It can be done as a 2D technique or a 3D technique. The time taken is slightly more. The advantage is you can compare with the opposite side, and that is something which we routinely do. So in chemical shift imaging, we use the phase encoding direction also for localization, which I was talking about. The phase encoding direction is one of the techniques. Gradient is applied for localization. Here in chemical shift imaging or multivoxel spectroscopy, we use the phase encoding direction also. The disadvantage is there could be a lower signal to noise and spectral quality for an individual voxel. So you get multiple voxel and you're looking at problems in each of them. And there could be a problem of shimming. So what is shimming? So whenever you are doing spectroscopy, and this is very important, there is a very good shimming is needed. Homogeneity of the magnet in that particular area is needed. Not only the magnet should be good, the area where you are going to do the spectroscopy is nicely to be shimmed, and this you might be knowing very well. And to homogenize that, you have a lot of techniques that we use. Water suppression, fat suppression is done, shimming is done to homogenize. And in human body, what is done is you have something called outer volume suppressions. So suppose you're studying a tumor at a particular area, the outer volume areas are suppressed so that you don't get too much of signals in from those areas, and to make that area of interest signals to be homogenized. Another important thing which is there in MR spectroscopy is the TE value, the time to echo value. This is very important as far as the radiologist is concerned, because certain peaks, certain metabolites that you are looking in the brain become prominent at a particular TE. If the TE is low, if you have a TE of 20 versus a TE of 135, you see the glutamate and glutamine are hardly seen when you use a time to echo of 135, but it is quite well seen in a low TE. Ionositol, not very well seen here, but at a intermediate TE. This is called intermediate. 135 is intermediate, and there is long TE, which is 240 and more. So that is not something which we routinely use. We usually use a low TE and an intermediate TE, so that we get most of these peaks. And also to differentiate certain tissues or differentiate certain metabolites, this can be useful. For example, lactate versus lipid. If you use a 30 millisecond TE, then both will be upright and you will not be able to tell which is lipid, which is lactate. You just do it at a higher TE, then you can see all the lactate will get inverted, but lipid will not be doing that. And again, when you do at a very high, then the lactate becomes upright. So this sort of changing can help us to tell what metabolite you are looking for. Then your voxel size. If you increase the voxel size, your resolution of the spectra and your peaks and all are going to be seen much better. Of course, it takes more time. If you go to a smaller voxels, then the baseline you see, and you don't see all the peaks as well. So if you want to see more peaks, more metabolites, larger voxel size, but you cannot use it always. There are very small pathological problems in the brain, tuberculous involvement in the brain and all can be very small. So you have to use a smaller one. Then the field strength is important. You use a 3 Tesla machine, your all these metabolites are much better seen. In 1.5, some of these metabolites might not be seen. And then you have some other things, which this is the Metabolite Map that you get in a multivoxel spectroscopy. And this is called a spectral map. And this is a black color metabolite map. You might be thinking, why do you want such a map? This is an NA map, N-acetyl aspartate map. This is an NA by choline map. Because these will show in very simple terms for a neurosurgeon in the areas from where the choline is raised or a particular metabolite it's more. And why should the neurosurgeon know it? The neurosurgeon will understand that this is the area where maximum membrane breakdown is happening. And because there is a maximum membrane breakdown, the choline is high in that area. So in the areas where the choline is maximum, you should go and take the biopsy. So such information is important. This is the short T brain spectrum. This is the amplitude here. As I was telling you, the y-axis is the amplitude and the x-axis gives the various metabolites. The Na represents the neuronal marker. The choline gives us the information about the membrane integrity. You see these values at a particular ppm is shown below here. So you get Na at 2.5, 2.6 here. Choline, you get at 3.22. And creatinine, phosphocreatinine, you get at 3.03. So all these occur at particular areas, and that is well visualized here. And then the same thing, myoinositol, which sir was telling about, is useful. It's a glial marker. Lipids gives information about necrotic areas in the brain. And glutamate is a very important, it's a neurotransmitter. GABA is a neurotransmitter. And Presence of glutamate gives information about toxic things inside the brain. So each of these chemicals or the metabolites in the brain gives information about a particular disease. These are the various diseases which can be picked up by looking at these metabolites. Now I'll just show you a few cases to describe it. Now this is looking something abnormal in the brain. Here again you can see it. Now these are then you do an MR spectroscopy of that particular area and you compare it with the opposite area. What you will see is the spectra is almost normal. Both the N-acetyl aspartate and choline are normal in this case. There is not much of an increase. In fact, there is some decrease. Now, this is what we usually do. We take in that lesion, in the pathology, in the tumor, outside the tumor, and then compare with the opposite side because we are doing multivoxel spectroscopy. And then we tell that whether it is a tumor, whether it is changes due to treatment, or whether it is a malignant or it is a benign lesion. So all these information which we give comparing with opposite side. So a very similar lesion as the previous. Lesion is the disease. What I am talking is lesion is the disease. If you compare with the previous, this also looks very similar. But here you can see the choline has gone up very high, which was not the appearance here. I'm just showing you again. Look, choline is in the lesion is small, but here the choline is gone up very high. So what you understand is the membrane breakdown happening in this tissue is more high, and therefore this is a tumor which is growing. It's more malignant. It could be more malignant. The other one is more benign. So that information we get. This is another case where you are looking at whether it is a tumor or a tumor-related changes. After radiation, there is something called radiation necrosis which is happening. Radiation-induced effect. And here you know that there is no membrane breakdown happening at all. Choline is very low. But here it is the necrosis which is happening. So that is why the lipid and the lactate which has gone up. So that information you get. Alzheimer's disease, which I told you earlier detection of Alzheimer's disease in the mild cognitive impairment stage is possible, looking at the myoinositol peak, which is actually starts increasing compared to the normal. And after some time, the neurons decreases in the brain. So it's an early marker, earlier biomarker for the disease of Alzheimer's disease. This was a very interesting case. Actually, we got a very unique spectra in this case. This is within a tumor. We are not expecting the N-acetyl aspartate. We got similar another case also. You see a very big peak. We actually did not know why such peak is coming. So at the time, the surgeon took this fluid from this tumor, gave it to us in a bottle, and then we went to the Pampady Institute, where they have a facility for in vitro spectroscopy. And then we saw that it was not due to the neurons, N-acetyl aspartate, but it was due to N-acetyl glucosamine and galactosamine. It was also the spectra which is very close. So in a mucus, we can find out this. So this particular tumor had a lot of mucus. That information we got. And actually this was published, and after that, this paper has been cited for many other, for detection of bronchogenic cysts or many other tumors also. This is useful. So this is again another review. The pus inside the brain can have a particular spectra in which you have a lot of amino acids, and this can be, and the succinate and all can be raised. This is brain worms. You would have heard about these worms inside the brain. Neurocysticercosis it is called. We don't call it brain worm. I just used it a simple term. And this neurocysticercosis sometimes can be slightly difficult to diagnose, but they give a particular type of spectra. It is common in tropical countries, India, Mexico, and all. It is quite common. Fortunately, in Kerala, it is not that common. And you get this particular peak, which is caused by the succinate. It is not the NA, but just close to that, there is another peak, new peak, which comes within this lesion, which is the succinate. This is a disease which happens in children, and it can be seen in some of the adults also, where it is called the Mela syndrome. And I don't want to describe more about it, but in this, there is a specific thing which happens. Lactate gets accumulated inside the brain, and that the lactate is being picked up. In patients who have cirrhosis of the liver, when it affects the brain, it is called hepatic encephalopathy, and there the glutamates increase in the brain. And when the patient is on treatment, you can monitor the treatment of the disease, the glutamate goes down. So another utility. So, and then this particular disease, a child comes with a large head, and then you do the MRI, you see a lot of abnormality around here in the white matter. It's all looking white. Actually, the white matter should be looking slightly darker. Here, the whole thing is white. But this can happen in many conditions. But in spectroscopy, when you do, there is this particular peak, and that is the N-acetyl aspartate peak is very much raised because there is an enzymatic deficiency. This particular chemicals gets deposited in the brain and it can be picked up. And we know that it is Canavan's disease. It's a disease called Canavan's disease. So there are so many uses of spectroscopy. We have also used something else, spectroscopy, for functional information. It's called a functional magnetic resonance spectroscopy study. Of course, this is a research study. Routinely, we don't do it in a clinical setup. A patient with mild cognitive impairment versus a normal person, we compared. And what we observed is, whenever a person is doing a memory function, yesterday, Dr. Senthil would have showed you about many functions. The memory function when the patient is doing, what happens is the glutamate increases in a particular area. But in patients with mild cognitive impairment, the glutamate might not increase. So this is called the functional magnetic resonance spectroscopy. Last few slides on diffusion tensor imaging, where you are actually understanding the white matter tracks in the brain. This is diffusion tensor imaging, and you have isotropic diffusion and anisotropic diffusion in the brain. And based on that, you can look at the white matter structures and the destruction or the abnormality of the white matter. White matter is the connecting areas in the brain. Suppose there is a language area and there is a motor area and there is a decision-making area. The decision-making area has to be connected to the language area, and the language area to the motor area for functions to happen. These are wires. If you see an electrical circuit, these are the wires. And these wires carry information of one thing to another area, from one neuron to another neuron. So these wires can be visualized by means of diffusion tensor imaging. And there are every wire has a different names, and these are called fibers within the brain. These are the fibers. This is, for example, the motor fiber, which carries the main movements of the body. This is optic radiation, which carries the visual functions of the body. So each area has a particular function, and you can do a tractography. Now here is a tumor here, and there are tracts which are going close to, and the surgeon wants to preserve these tracts so that after the surgery the patient does not develop a weakness. And so this is a DTI fiber tracking technique which is done. I'm just going to different techniques. I was discussing about MR spectroscopy, DTI fiber tracking is and diffusion tensor imaging is another technique. And another technique is the perfusion imaging, where you know how vascular, how many blood vessels are there in a particular area in the brain, which is again important. Tumors which are very malignant will have a lot of blood vessels, and tumors which are not very malignant may not have that many. So you can understand the vascularity of the brain, which is used by perfusion imaging. So finally, you have seen that we have so many techniques, but what the radiologist usually does in a clinical setup is he looks at the images, look whether there is normal things in the brain, everything structures are normal. There are areas which there are some brightness, abnormal blackness or something which are called the lesions, which is looked into. And then based on that, and contrast sometimes is injected, gadolinium is injected, and finally we come to a diagnosis. The problem of this is, it can be very qualitative. I tell that there is some amount of brightness. Another radiologist can tell that I don't feel there is much brightness. And that is where quantitative imaging is coming, and that is where the role of biomarkers are coming, and that is where the role of spectroscopy, which is a quantitative technique, Similarly, DTI, diffusion tensor imaging, and all these quantitative techniques are coming. So there is a need to develop more MR imaging biomarkers and quantitative methods. Of course, there is a problem. There is different machines gives different quantity. It is not like you measure your blood sugar. Wherever you go to measure, it's not going to be much different because the methods that they use is almost similar. But in MR, if you are using a 3 Tesla machine, a 1.5 Tesla machine, the sequences, the injection rate, everything matters. So there is a lot of variability, and a lot of studies are being done to now make it more uniform. There are consortiums created to make a quantitative MRI imaging. For example, in patients with Alzheimer's disease, memory decline, you can look at the volume of the brain, you can see the spectroscopic changes, you can have particular values for all these things and say that if these values are there, it is usually abnormal. So, so we are going from qualitative imaging to the quantitative imaging. You can get information of T1 relaxation value at a particular region, T2 relaxation time value at a particular region. So you put all these things together, you use artificial intelligence, machine learning also, and finally you get the diagnosis by quantitative methods. So this is where things are going, and I'm sure that this whole technology is going to help us much more in the coming days. Thank you very much.
Okay. Thank you, Professor Keshadas, for your excellent presentation. Now it is time for asking questions. So we can ask questions now. Or if they need any clarification, they can ask now.
Sir, I am not seeing any questions coming up.
No, there are some questions in the chat box.
So I have to open the chat box.
Yeah, please. Yeah.
So where is the chat box? I use this Google a bit less. So
okay. So
The right side top corner, Keshav.
yeah,
Right top
side.
corner. There's a chat.
Just near the list of participants. You can see the list of participants on the right side.
77.
Okay. Thank you. I have got it. I got it.
Okay.
So the questions here are, will subjects with tattoos also suffer the same fate as mascara wearing ones? Is there a workaround, or is MR not an option for such cases? Yeah, a very good question actually. Some of the tattoos are also can cause problems, and there have been cases where after tattoo patient had burns. Now it also depends on the chemicals that they use. So if you are doing it, you can ask the people who are putting it whether it contains iron containing things and all. So ensure about it and then only do it. I would say that better to avoid tattooing, but in case you are getting it done, do it in centers where they are very sure that what the materials that they are using and it is safe. But there have been reports. There have been reports by which tattooing has resulted in problem. So nowadays it has become a fashion. That is a problem. So we have to be careful and ask these to the people who are doing it. The next question is, can MR scans explain high and low intelligence in people and their IQ? No. MR cannot explain this. There are many other neuropsychological tests which are available by which you can actually, there is a whole list of studies which you have to actually task, which you have to do. You have to finish, ask these questions are asked, you have to do these answers to that, and from there you can tell this is the IQ of the person. This is called neuropsychological test. This is done in many of the neurology hospitals. This can be done. This cannot be estimated from MR. MR will just give an information whether the brain volume, for example, is decreased or compared to a normal population, there is a decrease or an increase. But that does not mean anything. Sometimes it might be just a normative standards. So it does not give any information about that. Great, doctor. How are the gadolinium biomarkers removed? Very good question again. How are the gadolinium biomarkers removed from the body? So nowadays there is an increasing evidence to suggest that this gadolinium sometimes get deposited within the body. Especially there are two types of contrast media. I was telling about gadolinium and then the chelating agent also. And some of these have a microcyclic shape, and some of them have a linear ones. If the gadolinium is not very stable with the chelating agent, they can be free and they can get deposited in certain areas in the brain, as well as in certain other parts of the body also. So it becomes a problem, and therefore nowadays, for example, in Europe, they have stopped the linear agents because the gadolinium becomes free and gets deposited, while the macrocyclic agents are much more safer. So removal of these things sometimes might not happen. Usually it has to be excreted from the body by our renal system, but this may not happen always, and especially it becomes free and starts get deposited. So it's a good question, and you can also ask your radiologist when you are going to get an MR ultrasound. MR contrast media being injected, whether it is linear or macrocyclic. What is the type of agent? And you can ask for a, if possible, a macrocyclic agent.
Just to add to it, nowadays most of the cases we ask for creatine level before we give the contrast.
Yes,
that has become a routine now because kidney has to secrete it. So
yes.
Even in the requisition form, it is written now.
Yes.
In AIMS, we have incorporated it.
Yes, sir. So creatinine is something which always we also look at before doing an MR, especially contrast if it is being injected. There is another question: why is gadolinium used as a biomarker? Any special reasons? Are there? No, actually, gadolinium causes the. So basically, you are looking at the contrast, isn't it? You are looking at whether your T1, and I was describing to you about T1 contrast and T2 contrast. This gadolinium can shorten the T1. T1 shortening can be produced. So there will be a change in the signal. Similarly, that is what is being picked up. So certain tumors or certain lesions, because in the brain there is the blood-brain barrier breakdown, these take up this gadolinium agents and then causes a change in the T1 contrast, and that is what we are measuring. Of course, there are other agents also. Iron, metallic agent, you know that iron is a contrast media which can be utilized, and you can use the sequences T2 star methods after injecting iron, but it has not become at all popular. People have started using it in Europe and in US also. It was mostly used for research setting. Even now you can see publications of people using iron, and I have several times asked why iron is not being used. It is quite a safe agent, but somehow it is not being used. Iron also gives a very good information about inflammation. Sir, you wanted to tell something?
Yeah, basically the complex element of gadolinium has a unique electronic structure compared to the other one. So if you put manganese or ferrous, then it more or less, depending upon even a small concentration, it washes out the signal. So you don't see it. Whereas this one promotes relaxation, but at the same time, it does not have any effect on the structural or the morphology of the thing. So it doesn't wipe out the NMR signal. Suppose it you use a long, high manganese, then it completely wipes out the MR signal. Then you won't even see the images sometimes. So it's very difficult. That is because of this unique electronic property, it is prepared compared to the manganese or ferrous.
So this is another question. Of course, there are a lot of people who are interested in brain. The question is, in the case of photographic memory persons, is there any change in their brain hemispheres compared with ordinary people? Now, there are a lot of research studies going around the world about all these things, looking at functional MRI as well as looking at the volume of the brain by means of voxel-based morphometry and various other techniques. So studies are happening, but these are all research studies. I don't know any research study which has looked into photographic memory people and said that this hemisphere is more stronger or this hemisphere is lesser. But there are connectivity studies. I was showing you, there is something called functional connectivity now, which is coming up, and which we can study actually the brain functions much better. I'm sure Dr. Senthil would have discussed about it yesterday. These things might help over a period of time. At the moment, all these are in research and usually not much use in a clinical setting. So in India, actually, these are called cognitive studies. These sort of cognitive studies are happening much lesser. Indian Institute of Science probably, then institute in Allahabad, Cognitive Sciences Institute is doing, some in IIT, Delhi. So groups of small people are doing it. Otherwise, in a medical setting, it does not have much applications.
The left-right asymmetry is known already. And there was one report which says that some neurobiochemicals may be different in the left and right, but it has not been proved by other groups. So basically.
Sir, so I think that was the last question, and thank you very much for it was really nice to interact with students. I hope my only thing is that my talk and the talk of Dr. Senthil as well as Professor Jagannathan, all these were very applied, and to what extent our students enjoyed, I am not very sure. But I hope such talks are very useful in our MRS society. It will be very useful for students so that they can take up these research into applied medical field also, whatever they
I just would like to add, yesterday there was one question after the talk, was that what is the role of a chemist in it? Some participant was asking. I said, I just answered to him saying that I'm a physicist. So basically, so everybody can contribute. And I came into MRI from a physics angle, and whereas Keshav from a radiology angle came to physics and then understood that. So when I in fact joined AIMS in 1993, the first 1 year I spent in anatomy class with MBBS students, so that I learned about it. So chemists have contributed. In fact, most of the Nobel laureates, right? Richard Ernst is a chemist. Paul Lauterbach was a chemist. Mansfield was a physicist. And Kurt Utrecht, who did the 2D NMR application, is a chemist. So chemists, physicists, and radiologists have a lot of role. Unless we join together, we cannot do that. In fact, the ISMRM is the Society International Magnetic Resonance for Medicine, which we started mainly because to bring everybody into one platform. And I omitted completely both of us, the technologists and the engineers. They have played a very, very big role in that. And without the engineering, technology, physics, chemistry, and radiology, nothing can move in MRI. It's a very vast field, and what we are given is an iota of what applications are. So many things I still don't know. I spent 30 years, but I don't know many things still. It's a very ocean. NMR is a ocean, and this one is a separate ocean by itself.
Yeah.
Thank you, sir. Thank you, Keshav sir, for this wonderful talk. Paulson sir, can you conclude?
Yes. We are listening to an expert in MRI for the last 60 or 70 minutes. He had discussed different aspects of MRI, including image generation, 3D sequencing, and most importantly, the safety issues. And he has already discussed the various case studies in clinical studies we have done. And on behalf of the participants and the organized committee, I thank once again Professor Keshav for your excellent presentation. Thank you, sir.
Thank you, sir.
Thank you, Paulson sir, for chairing this session. Thank you, Keshav sir, for the wonderful talk. It was great. I have lots of questions, but I thought I'll stop. That is why I didn't want to ask here. but maybe when we meet or somewhere, I can. And also Janan sir. Janan sir, you can always visit Trivandrum. We have a wonderful guest house in Isar. It's a very nice place.
Yeah, new place you have moved in, right? But can you take care of COVID-19? Then I
that is not in my
yeah, that is the one. No? See, I used to travel. When I joined here, the first thing I asked the VC is that I travel a lot, and he gave permission. So I used to travel twice a week to Delhi and other places. Now suddenly, from March 18 onwards, I haven't traveled at all.
Anyway, Keshav sir is also here.
Start working for any first venture now. Things.
Yeah.
It was nice, and thanks all of you, especially the organizers, to make at least, I think 40% of this symposium was on MRI, more or less. And I think that would have given the participants sort of understanding how this MR has revolutionized the field, and MR has no boundaries. See, that's what it is. It has no boundaries at all. I mean, you name it, you have the application. Material science, agriculture. I have done some agricultural plants when the first biospec came to us, animal MRI. I published 6 papers, and then it fetched me at Japan to Casper. So enormous amount of applications. Once you know the basics of it, it's very easy to apply. And thanks for the organizers to make it as 40% of the 100% NMR. Because in most of the NMR conference, we have a very minuscule representation of MRI, and many people don't even appreciate MRI from a regular NMR. Many people are not aware. They think that it's only for structure and characterization.
No. I mean, I'm always fascinated with MRI because that's the direct relation to.
And the beauty of NMR is that MRI started from NMR, and many things that are used in NMR.
I mean, it's the technical part is very easy.
Reverse is happening. The gradient spectroscopy that you do on mice and other things for even 2D to shorten this one all came from MRI. So the knowledge of flow from MR to MRI and MRI back to NMR is something very fabulous. One has to really appreciate that, how the applications are.
Yes, sir.
And one more thing I want to tell for the youngsters is that whenever you want to do some work, make a plan and have something which is very useful. Because when I sit in the committees, especially chair of devices and diagnostics in DB2 and BIRAC, they don't have clinical collaborators. So we always tell them, don't invent or do any fabricated, manufactured devices and look for application. Instead, you talk to clinician and find out what they need. Then start developing the device. Then it becomes more useful. More useful. Or else it will be an device that will be looking for application. So rather than it should be the other way. So it's very nice to see a lot of youngsters participating. And thank you very much.
Thank you, Jagannathan sir and Keshav sir for wonderful talks. So I think we can conclude this morning session, right?
Yes.
Thank you all. Yeah, we will meet at 2 p.m. Our afternoon session will start at 2 p.m. and the Google Meet link will be open at 1:45 p.m. So thank you all.
Thank you.
Thank you, sir. Bye.